All right, welcome back, everyone. So we're going to now take a few minutes and look at still our innate immune system, but now in particular, what we're going to look at is the second line of defense, right? So remember, the first line of defense, this was think barriers, right? So think skin, mucous membranes, and then some other things, so like the mucociliary escalator, stomach acid, saliva, things like that. So the second line, again, still non-specific, right? So still not. Sometimes you may even see the term non-discriminatory, right? So the cells don't discriminate against a particular type of pathogen. They just know it's foreign. We're going to attack it, right? So in this case, now we're looking at these various specific cell types that we're going to look at. So selected immune cells, some different proteins that we're going to look at, and then things like inflammation and fever, which we're all likely familiar with, right? So our cells that we'll discuss, these are basically our immune system cells other than largely those other than lymphocytes, right? So things like neutrophils, macrophages, dendritic cells, these are all phagocytic in nature, right? Meaning they cellularly engulf cells or pathogens and then present them to other cell types, right? So these dendritic cells are an example. These are antigen presenting cells that will engulf a pathogen and then present a portion of that pathogen called the essentially the antigen, if you will, to our specific immune system, so to our T cells, right? So this is necessary for initiating adaptive immunity, right? Adaptive immunity being the specific immune system, right? Our adaptive or our acquired immune system, right? So we'll have separate lectures and separate discussions specifically on the adaptive side of the immune system, right? So this graphic here is simply showing some of those different phagocytic cells, right? So you perform phagocytosis, right? Remember, that is a type of endocytosis. That engulfed pathogen is in a vesicle that's called a phagosome. This phagosome is going to fuse with a lysosome, and that forms a phagolysosome, right? So essentially, the chemicals and the enzymes in the lysosome are going to fuse with the phagosome, break down the pathogen. So in this case, some type of bacteria, some type of virus or something, destroy that agent, and then that remaining portion is going to be exocytosed, right, and released out of the cell, right? So you also have some other cell types like basophils and mast cells. These circulate in the blood. They can also reside in connective tissue and mucosa as well. And the main thing with these is they're going to be secreting and releasing various types of chemicals, right? And these chemicals are going to have effects on the cardiovascular system to some degree, but also they're going to obviously have a major role in our immune system, right? So the two main chemicals that are released here are called histamine and heparin, right? So these histamine and heparin molecules, and you can see them here, these are acting on blood and blood vessels, right? So histamine is a very potent vasodilator, right? So essentially what's happening are the blood vessels themselves are going to increase in size, increase in permeability, vasodilate. And what this does is this allows more blood to flow into a particular area, thus bringing more immune system cells to the potential site, the infection site, right? The other molecule, something like heparin, heparin is an anticoagulant, right? So it's going to minimize blood clotting, which again is helping to promote the flow of blood toward the infection site, right? And then you have things like eicosanoids. These eicosanoids are helping with inflammation, and we'll have a whole discussion on inflammation coming up here in just a moment, right? Another cell type is called a natural killer cell. These are, excuse me, these are essentially types of lymphocytes, right? They're kind of derived from the lymphoid line. And these are performing something called immune surveillance. So they're kind of looking out for unhealthy or unwanted cells and then initiating responses against those. And the way that they do it is through something called perforins and granzymes, right? So perforins and granzymes, essentially think of these as, think of the perforin as something that forms a opening or a hole in the membrane of a cell. So think the word perforin is like perforate, right? So you're creating this opening or this hole in a cell. And then the granzymes enter the cell, enter through these new openings that were formed by the perforins, and then go in and they initiate and induce something called apoptosis. And so apoptosis is essentially just programmed cell death, right? And so again, these are unhealthy cells slash unwanted cells that the body is going to get rid of. So maybe things like cancerous cells or potentially pathogenic cells, right? An additional cell type is one called an eosinophil, right? So eosinophils are one type of our leukocytes, right? So recall you have your granulocytes and your agranulocytes. This is a granulocyte, right? So meaning it has granules. Remember, it ends in "phil," and these especially increase in number in response to allergies and parasitic infections, right? Those are kind of the main things that they proliferate in response to, right? But these are also phagocytic cells that involve or that are involved with that process of phagocytosis, bringing in various types of pathogens, foreign antigens, unwanted cells, things like that, right? So let's stop here. This is a relatively short part, and we'll pick up in our next part, and we'll begin talking about and looking at some additional cellular processes involving proteins, things that have to do with combating viruses, and then we'll get into talks about inflammation and fever.